I'm Renu Kumari from Semantic Climate. So today I'm going to talk about the knowledge extraction from the PDF papers, and that is the first step of the automated literature review. So to begin with, what is like I'm from the Semantic Climate. So Semantic Climate is a, like more or less 6 year old organization, and it is a team of interns. They are joined online or offline for the 6 months or less than like 6 months, and then they work under the guidance of mentors and they develop the tools for literature extraction or knowledge extraction. So we also organize the workshops and hackathon and also go to the schools to give the knowledge about the climate. So so why the literature review matters? So when we do the literature review, it is all the foundation of any research. Like when we go for the PhD or any writing thesis, so we go to search the literature and then do the review. And also we wanted to understand the existing knowledge, what are there in the literature already, and we wanted to identify the research gap, and also we wanted to avoid the duplication, so that we work on the new or novel data. So for that, we are going for the literature review. So the challenges we are facing with the manual literature review, that is, we are getting the exponential growth of publications each and every day, and these publications exist in mainly in the PDF format, and the PDF is always is not a machine readable format. So and it is also the time consuming process when we go for the manual review. So Semantic Climate team has developed some tools to do the ALR, that is the automated literature review. So in the automated literature review, there are many methods of doing this literature review. The first method is we are explaining the named entity recognition. The other methods are the summarizations and the key phrases extraction. So today I'll talk about the named entity recognition from the scholarly literature. So what are the named entity recognition? It is the name of the species, genes, or protein, or name of the locations present in the research article. So we extract using the toolkits from the research articles. So the workflow we are following is when we go for any literature review, so like we give the query to the tool, that is the Pygate paper, and then Pygate paper, we give the query, and then Pygate papers go to the open access repository. For example, European C, it can also go to the Open LX and then give the research article. And then after Pygate papers, it gives the different kinds of formats, like we can download the PDF and it gives the XML and HTML format of the papers. And we can also create the data tables, that is the semantic index of the whole literature we have searched using the MLIB tool. And then this third tool developed by the doc analysis is used to get the named entity recognition from the literature. Hmm. MLIB doc analysis. Location. Hmm. So here in this workflow, we are directly searching, like we are starting from the searching the query, like any art, like we are wanted to search on the drug discovery or any natural product or any about the plants or anything. So we give directly the query to this Pygate paper and it will search the papers from the open access repository. Pygate, Pygate paper. Hmm. Hmm. Like like we are searching for the Google. Hmm. This one is for creating the semantic index. Hmm. Like I'll tell you in the further slides that how in the hands-on we will do. So the first tool is the Pygate papers. So it was developed by the Ayush Garg, and he was also the intern with the semantic climate and under the guidance of the Peter Varela. So this is the tool like similar to the Google. We go to the give the query and then search the PDF and go to the each and every paper and then do. But in this Pygate paper, we just write the query and then it will number of. Okay. There's no. Yeah, kind of this. Yeah. Okay. Okay. So okay, fine. Huh? So basically this tool give query to this tool and then it will search the papers and we get the papers in the form of PDF also if it is available openly. But generally we are taking from the EuroPMC and all the articles are available there. And also we are taking from the OpenLX. If PDF is available on the OpenLX, then it will give. So it is based on the API we are giving for searching the tool. So, so this give the papers in the HTML format, XML formats, and the PDF format. So after that, the whole literature we give to this, like they have the unique features also. Like we give the dates also, like you can get the papers from this date to like this date to end date, and also give the multiple queries in a file and give to the Pygate papers. The second tool is Emily, where it creates what the research article we have searched. It creates the data table. Data tables give the summary of like what are the titles, what are the author names, what are the DOI, and what are the like a brief abstract of the whole literature we have searched in a single format. So third tool is the doc analysis. It will do the sectioning and NLP and also the NER based search. So so how we are giving the query to the Pygate papers, it is a command line tool. So we give like here the query we have given drug discovery and the medicinal plants. And we have given the format like XML. You can also put the like minus PDF. So it will give all the PDF related to that query. And we can put the number of paper limit. Here I have given the 50, but you can like 100 or more than 100. It will easily like save the papers and dates and then you can give the output directory name and it will save all the query results. So here we are getting, I have given this query and I'm getting this drug discovery output directory where all this paper from the EuroPMC has been saved and we are getting the full text.xml because we have given the XML format, but you can get the full text.pdf also if you are given the PDF options to download. And we are also creating the data tables. So the data tables will look like this. Here we are getting the ID and the DOI of the paper and the title of the paper and authors and where it is published and in which year it is published and also the brief abstract So that when whatever the query we have given, is it relevant to our search or not, so that we can further move to the analysis, or you just rephrase the query and then search again. So this table will describe all the things. You don't have to go to the directory and see the what paper it is coming or something. Now, after that, we are applying this doc analysis to extract the named entity present in those papers. So I have given the drugs and disease and the location, but we can also search for the other, because some dictionaries-based search are there, supervised analysis are there. So we are extracting those informations from those papers. So, and now we are creating the knowledge graph, whatever we have searched on that query. So in this knowledge graph, you can see that this paper, blue color represent the research article. In this research article, this purple circle presents the country name or place name. So these paper contain these many numbers of places. And also the green color represents the drugs mentioned in drugs or chemical. And gray represents the disease. So it graph shows that how this paper is connected to these countries and the disease and the chemical. And all these nodes are clickable, and it will go to the Wikidata. You can get further information to each and every node. So the tool we are using is running on the Google Colab. So the Google Colab is like a simple Jupyter e-notebook, and here you don't have to set up anything, and it's just like you need a Gmail account or Google account, and it will work accordingly. So here is the interface of the Google Colab. You can change the title of this Colab notebook, and also here is the two code cell and the text cell. In the text cell, you add the documentation about the whole system, and in the code cell, you write the code for running the Colab notebook. Here, it is all the files are stored, output, whatever coming in the Google Colab, and then here you can also upload your own files in the Google Colab. So before using any, like if it is your Colab notebook, then there's no need to save it in your drive. But if it is shared by others, so first you have to save in your copy in a drive, so that you can change and save all the changes according to your need. And sometimes it depends on like running the code, you require more memory and space, so you can change the runtime like a CPU to GPU. So with this, I'd like to say thank you. And the hands-on session will be done tomorrow. Will be done by the Preeti. So thank you, everyone.